Dear students, last time we had studied osmosis and diffusion. As I had mentioned in the last lecture that the diffusion is of two kinds. One is simple diffusion and facilitated diffusion. And regarding facilitated diffusion, I told you that it requires a special carrier protein called as permease to function. And it is a bit rapid than simple diffusion. Now let us see what is the mechanism of action of this facilitated diffusion. You can cite in the slide that the solute is outside, depicted in form of black colored circular picture, and it is just taken up by the permease. Now after formation of permease and solute complex, the conformation, as you can see in the slide, that the conformation of permease got changed, and after changing the conformation, the solute is thrown inside. Such a manner of transport, it happens in human erythrocyte when it transports glucose, and it is transported by glucose permease, and this is the transport of glucose inside the human erythrocyte is a facilitated diffusion, and it is guided by insulin. Then uh, the lines following this is of lesser importance to you. Then come to the factors affecting the diffusion. Another is band 3 polypeptide and anion exchange permease protein. It transports chloride and bicarbonate ions into erythrocytes to maintain the electrical neutrality in the RBCs, and this is also a facilitated diffusion, and it helps transport of CO2 also in form of carbonic acid. Factors affecting diffusion. Permeability and thickness of membrane is the reason factor affecting rate of diffusion. Lipid solubility, number of pore channels, temperature, molecular weight of solute, and diffusion coefficient. Then most important way of transport is active transport. It is the movement of the solutes or substance against the concentration gradient. However, naturally no substance moves against the electrochemical gradient, that is, uphill movement is not affected until energy is involved in it. In active transport, when no energy is used, no uphill movement will occur. I beg. The active transport is required because many a times in our body we require large concentration of some ions, even if such ions are less in extracellular fluid. This is true for potassium ions. Low concentration of some ions required to be maintained in ICF even if such ions are more in extracellular fluid, and this is true for sodium ions. So sodium, potassium, H ions, chloride ions, calcium ions, Fe ions, sugars, most amino acids, they are transported actively. That means there is involvement of ATP in it. Energy is expanded in this kind of transport. There are two kinds of transport: primary active transport. In this transport, as usual in active transport, it is transported against the concentration gradient depending upon the carrier protein. But here the carrier protein functions somehow different from the carrier proteins which I had mentioned you in facilitated diffusion. For it is capable of importing energy to the transported substance through ATP hydrolysis. This ATP hydrolysis capacity was not afforded to the permease of which was involved in the facilitated diffusion. But here you can see the carrier protein has the capacity to hydrolyze the adenosine triphosphate, which are the energy molecules. The mechanism of active transport: sodium potassium pump is involved, presence on surface membrane or intracellular membranes, transpose sodium out even though its concentration outside is high, transport potassium inside even though its concentration outside is low and inside is high. So this sodium potassium ATPase is a carrier protein with the capacity to hydrolyze the ATP, that means having the ATPase capacities. It is a dimer having two polypeptide chains. Polypeptide one first is glycosylated, second one is non-glycosylated. But this polypeptide 2 has the peculiarity in the sense that it has a catalytic site for ATP hydrolysis. And as I mentioned, transports 3 sodium out and 2 potassium in. Look at the sodium potassium carrier protein has 3 receptors. You can see in the slide that inside you look at the ICF. It carries 3 sodium binding site where 3 sodium binds and they are transported actively to outside, and you can see in the at this ECF that 2 potassium they are carried in and transported to the ICF. Because the sodium potassium ATPase carrier protein has 3 receptor sites for binding sodium on side protruding inside, whereas 2 receptor sites for binding potassium on side protruding outside of the cell. And we had studied the integral membrane proteins, and this is also an integral membrane protein. The inside portion adjacent to sodium binding site has ATPase activity also I had mentioned, and this differentiates this sodium potassium ATPase carrier protein from the permease which was involved in facilitated transport. When 2 sodium binds on receptor sites protruding outside and 3 sodium on receptors inside, the ATPase function of the protein becomes invoked, and this this activation causes conformational change in sodium potassium ATPase carrier molecule, like the permease which was involved in facilitated transport. This extrudes 3 sodium out and intrudes 2 potassium inside. So students, another is secondary active transport. It depends upon the chemoosmotic energy, and in this although this this may not be in the scope of your studies, in this energy used in ionic gradient is utilized, and you just need to remember only 2 examples. That glucose transport system of intestine is the kind of secondary active transport in which electrochemical sodium gradient is utilized for secondary transport of glucose into the cell against the concentration gradient through glucose carrier. And there is glucose sodium co-transport. Through primary active transport, the sodium is kept low within the cell by excluding sodium out against the electrochemical gradient. This is utilized for glucose sodium transport. Both binds to transport proteins. When two molecules move in the same direction, such a transport is called a symport, whereas when the 2 molecules moves in different directions, that such a transport is antiport. The example is lactose permease system in E. coli. You will read this thing in the higher classes when you'll study the lac operon system. So these were the functions which are performed by the plasma membrane, that osmosis, the diffusion, the active transport. Another function performed by cell membrane is cell recognition. You might have seen that blood transfusion or organ rejections, incompatible blood transfusion is caused by the uh, proteins glycocalyx on the RBCs, and this sialic acid is involved in cell recognition. Mammalian leukocytes recognize foreign cells. Spleen macrophages can differentiate between healthy and worn out RBC. Spleen is not going to kill our normal RBCs because it has the capacity to recognize, its macrophages has the capacity to recognize the healthy and worn out RBCs. Our leukocytes, they do recognize foreign cells and they produce antibodies, and macrophages, they eat up the cells which are foreign to our system. Cell surface carries both glycosyl transferase and glycosyl receptors, which are oligosaccharide. Transferase of one cell bind with specific antigen of other molecule in this, and a complex is formed. In addition of monosaccharide sugar residues to receptor chain brings dissociation of ES complex. Cytosis. Then if we place amoeba in water into which carbohydrate has been added, no pinocytosis will occur, and if protein is added or amino acids are added, it involves pinocytosis. The outcome is that the proteins are catalyst for pinocytosis. Exocytosis is the reverse of endocytosis, and here the vesicle fuses with the plasma membrane and releases the content out, and it serves as the means of egestion or extruding the undigested material from the cell. And this exocytosis is the process by which many hormones, they are secreted out of the cell. Then chemoreception. Protein bounds on plasma membrane are believed to serve as a receptor for taste and smell in mammals. Transmission of nerve impulse, very very important. Transport of sodium inside and outside to potassium outside is affected by plasma membrane. They are so very important for the conduction of nerve impulse and carrying forward of action potential. So students, this is all in today's lecture. In the upcoming lectures we'll study cell junctions. Thank you so much.